But the question was, can we detect this in real time? Can we determine if we are getting amyloid beta fibrillized or not? Well, actually, yes. If you put this in an assay in which you have monomeric amyloid beta, and this is not trivial to do, but you have all completely monomeric amyloid beta at the beginning, you track the fluorescence of ruthenium bis BPD PPC, and you see that you get nothing for the first 100 minutes, and then suddenly you get a jump in the fluorescence. That jump in the fluorescence is due to the formation of small seeds of amyloid beta. This region here is what we call the lag phase, and people used to think that nothing is happening, but proteins, especially peptides, are very dynamic structures. So this peptide is kind of like playing, trying to fold itself in different conformations, and at some point, 4 or 5 of these peptides learn how to create a very small fiber. They teach themselves how to create a very small fiber, and then that fiber gets rapidly elongated, and it is the same as having a supersaturated solution of a compound and throwing a crystal in it. It immediately crystallizes. So here it happened exactly the same. Immediately you get fibrillization until you reach equilibrium because you don't have any more monomers to form fibers. I mean, if you take an aliquot from this point and do a TEM, then you get all these spaghetti-like structures. That is amyloid beta, fibrillized amyloid beta. But the question is why these things bind to amyloid beta, and we did a lot of experiments to unravel these. These are binding experiments. We determined that the binding constant, or the dissociation constant for these ruthenium complexes to amyloid beta is about 2.1 micromolar. The binding constant is, of course, the reciprocal of that. It's 4.8 times 10 to the 5 molar. It is comparable, although a little bit smaller than DNA, but this complex can intercalate in DNA, but they cannot intercalate in amyloid beta. And I'm going to show you in a second why. But anyway, we did other experiments like trying to get the stoichiometry of ruthenium complexes per amyloid beta monomer. And for that, you can do what we call a job plot, in which you change the mole fraction of RuBP and amyloid beta at the same time. And what you get is a curve with a maximum, and from this maximum, we get that there's about 2.6 amyloid beta monomers per ruthenium bis BPD PPC. This is an experimental value, so remember that 2.6 A beta monomers. So what else can you do? Well, you can do theoretical computations, and we actually did with some collaborations at the University of Miami. And what we found is that there's a hydrophobic cleft between phenylalanine 20 and valine 18. This hydrophobic cleft propagates throughout the molecular axis of the molecule. Actually, amyloid beta is very efficient. When it folds and forms the fiber, it hides the hydrophobic residues in the internal structure of the fiber, basically building the fiber backbone, and project the hydrophilic, which are the green ones, towards the water interface. But it can do so much, right? So at some point, there's some amino acids that are hydrophobic but still exposed. So the DPPC ligand, which is very hydrophobic, kind of like interact with those amino acids very, very tightly. And if you count, I'm gonna help you to count here. This is 1 amyloid beta monomer. This is 2 amyloid beta monomer, and it takes a little bit of the other one. So about 2.4 amyloid beta monomer from the theoretical model. We kind of like match very well with the 2.6 amyloid beta monomers from our job plot. So that's also very nice. So finally, I was telling you that ruthenium bis BPD PPC have a very large lifetime, about 185 nanoseconds in comparison with thioflavin T. So we can use that to do things that you cannot do with other fluorescent probes. For example, sometimes you want to test if compounds inhibit amyloid beta fibrillization. The problem is that the compounds that you want to test are fluorescent too. And many of these are polyaromatic compounds. So polyaromatic compounds have very strong photoluminescence. You can also use thioflavin because they will completely overwhelm the fluorescence of thioflavin. Actually, they completely overwhelm the fluorescence of ruthenium bis BPD PPC as well. Let me show you. We put rhodamine B, which is a standard fluorophore there, to see if rhodamine B can inhibit amyloid beta fibrillization. So before fibrillization, it looks like this. After fibrillization, it looks like this. So rhodamine B is completely dominating the spectral characteristic of the system. So you see a little bit of increase, but that is nothing. You basically don't have any resolution. So this is what you expect to see. This is in buffer. So you don't have any rhodamine B there. So what we did was some time-resolved studies, and we determined that the best time gate to do our experiment is here. So we're going to do that. This is our experiment, again, in buffer, just so you know what do you expect to see. And I'm going to show you now this. This and this are exactly the same sample, but this is using steady-state fluorescence spectroscopy. This is using time-resolved spectroscopy. You can see that now we have filtered most of the fluorescence from the ruthenium complex. You get a little bit there, but remember that rhodamine B was in a big excess here. So we really can see now a really good change in the fluorescence from non-fibrillar to fibrillar amyloid beta. And the question is, can you do it in real time? And can you tell me if rhodamine B inhibits fibrillization or not? And the answer is yes, we can. If you do it in real time, using steady-state fluorescence spectroscopy, you basically get nothing. Using time-resolved, now you can see a very nice sigmoidal curve that shows the real-time fibrillization of amyloid beta. But if you remember the first curve that I showed you that didn't have any rhodamine B, it started growing at 100 minutes. This one started growing about at 200 minutes. So the growing of the amyloid beta has been delayed. So actually rhodamine B actually inhibits amyloid beta fibrillization. But it's something that you could never have been able, an experiment that you would never have been able to do using thioflavin T or any other fluorophore, just using long-lived fluorophores and time gating. So we can do other things, and it might sound cruel, but these mice have been transfected with the gene of Alzheimer's disease. Well, with the gene of amyloid, the amyloid precursor protein, or APP. So they develop all the symptomatology of people with Alzheimer's. So you can take their brains out, you can slice those brains, and then you can stain those brains with our compound, ruthenium bis BPD PPC. And if you do that, and this is optimization in different medium, water, glycerol, and Vecta shield, which is a polymer. These things that you see there, these are amyloid beta lesions in the brain of a mouse. So our compounds actually not only can stain synthetic amyloid beta, but they can stain amyloid beta ex vivo in brain slices. Another thing that we can actually do is that we decided to try other proteins that also aggregate in fibrillar structures. And this molecule, protein, alpha-synuclein, is associated with Parkinson's disease and not that much with Alzheimer's disease. So this molecule also is an intracellular protein. Amyloid beta is an extracellular protein. But both aggregate, both form amyloid structures. And we basically decided to move from the bis BP to the phenanthrolene, just because the phenanthrolene has a larger binding constant, and also because its fluorescence is more intense. So for cellular studies, it's better. We did real-time, TEM, AFM. It just worked beautiful. So I'm going to show you now the cellular work. So I would like to show you, it doesn't look very well, but H4 cells, I went out of juice, I think. H4 cells are basically cells that don't overexpress alpha-synuclein. These cells overexpressed alpha-synuclein with alpha-synuclein, and besides that, we have a GFP protein fused to alpha-synuclein. In any case, we injected our probe, ruthenium fen DPPC, on treated cells, don't display any photoluminescence. Treated cells with MG132, which is a drug that inhibits proteasomal degradation, promote protein accumulation, more alpha-synuclein inside the cell. If you look here, you might be able to see some red things. Those red things are amyloid beta. Well, not amyloid beta, alpha-synuclein deposits, or amyloid deposit of alpha-synuclein. How do we know that it is alpha-synuclein and something else? Well, we have this cell line that has alpha-synuclein fused to GFP. So again, on treated cells, don't display any signal, no signal. But when you add the MG132, then you see red signal here. You see green signal here due to GFP, which is fused to alpha-synuclein. So anywhere that there's GFP, there's alpha-synuclein, and you get very good colocalization. So we know that we are actually staining alpha-synuclein without any problem. Finally, the question is, we have been showing all this time gating technology. Can we do it in the microscope? Can we do microscopy using time gating? And the answer is yes. We can do it using fluorescent microscopy, or what we call FLIM, fluorescent lifetime microscopy. So what you're seeing here, although they look colored and intense, is basically a lifetime image. These spots were done by lithography of amyloid beta protein. And basically, we created this pattern, and this is the rice owl. So it says right out there. So we also created this pattern with that lithography. But the rest is just amyloid beta protein. And what you can see there is that the background basically don't have any lifetime. It's a very short lifetime. The one that have amyloid beta with ruthenium BPD-PPC have a very long lifetime, 450 nanoseconds. The same with the owl. It just looks cool, but this gives you all the information. In any case, all this research was performed in Rice University in Houston. This is the main building of Rice University. And this right here is the city of Houston. And this is my laboratory, the second floor of Del Butcher Hall. And this is Houston, Texas Medical Center, one of the biggest medical centers in the United States. So many of our collaborators are actually there. And I would like to thank the people that actually did the work or the job. Nate Cook, Kiwei Huang, Abhishek Saha, Victor. Of course, this girl. Where is she? Marcia. And outstanding collaborators like Rajiv Prabhakar from the University of Miami, Laura Segatori from Rice University, Joanna Jankowski for Baylor College of Medicine, and the people that give us money, NSF, the Wedge Foundation, and Rice University. Thank you all for your attention.
My question is, for the detection of NKO, would you use the iridium complex? Could be other metal instead of iridium there? Osmium or ruthenium, for instance?
Yeah, we have thought about that. The imide group, it is actually responsible of switching out the fluorescence of this group. So basically, you could put in theory other metal centers there, and if you actually it would be very cool, because if you change the metal center, then you will change the electronic transition. You will change the color, and you will change also the excitation. You will change the Stokes shift. You will change the lifetime. So all those things are things that are interesting to us. But yeah, it's tunable. You can. What really happens is that when you excite the metal complex, the electron is transferred immediately to the imide group. When the imide group reacts with the cysteine complex, then those molecular orbitals are not available anymore. So the excitation needs to go directly to the phenanthroline. So theoretically, it should work with any metal complex. We haven't tried with many, but at least for ruthenium and for iridium, it definitely works.
I was just wondering how toxic are those compounds, and if there is a possibility that in the near future it can be used as probes for human treatment or for human detection of those.
That's an excellent question. Let me see if I can go back a little bit in my presentation. Oh, well, I'm going forward. So if I go back at least, wait, sorry mouse. There. So the thing about metal complexes, right, is that they are electronically charged. So the ruthenium complexes are +2 charged. The problem with in vivo imaging of humans or animals, because what we're moving now towards is to image a living mouse, is the charge of this compound. It is +2 charge. So we have something that we call the blood-brain barrier that prevents charged molecules to reach the brain. But the good thing about these compounds, we think, is that it has so many hydrophobic surfaces that they might actually trick the blood-brain barrier, might be able to cross. But what we are doing actually is that we are making these ligands more hydrophobic, and we are putting phenyl groups here in order to increase the lipophilicity of these probes in order to reach and pass the blood-brain barrier. Another thing that we are doing right now, and this is part of my NSF proposal, is modifying these ligands so we can change the absorption of these complexes that now is 445 nanometers. So we are talking about blue light. So blue light is not very good for in vivo fluorescence because our skin absorbs blue light very well. So we are trying to move this excitation about 550 nanometers, in which our skins start becoming transparent. So we are right now modifying these ligands so in order to shift to the red the excitation. The emission is good. 620 is good for emission, but we still need to work a little bit on the excitation. That's part of our current efforts. But yeah, one of the things that we are very interested in see if we can actually image amyloid beta formation in living animals.
What do you mean when you say hybridization, and why do you do this in your experiment?
All right. Maybe I didn't explain that very well. But when you, well, let me go. This is a small image of a fiber. So when I mean hybridization is that these proteins start to stack one over the other, forming a very long fiber. Let me show you the fiber. No, let me show you one of them. But basically, that is what it is. So it's you start stacking proteins over proteins over proteins over proteins over proteins, and what you actually get is like a long rope of proteins. And why that rope is important? Because when we all have amyloid beta proteins in our brains, children, 2 years old, newborns, they all have amyloid beta in their brains. This protein is ubiquitous, it's everywhere. But the problem is that if you have it in the monomeric form, you don't have no problem. The problem is when this protein transitions from the monomeric form into that aggregated form that I was telling you, that rope. That transition is associated with the beginning of Alzheimer's disease. So why we want to detect that? Because as I was showing you before my computer decided not to work with me, we can track that process in real time. So we can put drugs or molecules there and check how that real-time hybridization changes. If now it takes longer times to the proteins to aggregate, then we are delaying the aggregation. We are inhibiting somehow the formation of amyloid beta aggregates. So that kind of things we are very interested in doing. and developing probes that can do this in environments or with molecules that generally you cannot do with other probes, that's actually what we do.